Hi, we are the group 3. Today, we'll be discussing about 3 things that are essential to our lives. First is the cellular respiration, which will be discussed by Andrea Centena. Second is the photosynthesis, which will be discussed by Gerald Dalipe and Rafael de Belen. Lastly, the fermentation process, which will be discussed by Cedric Cruz and Divine de Vera. Let the discussion begin. First off is the cellular respiration. It occurs when we play guitar, when we walk, or when we run. But how does it work? Cellular respiration happens in mitochondria, which is a part of a cell which is part of our body, and it occurs 3 stages. First stage is the glycolysis stage, where chemical energy is harvested by oxidizing glucose to pyruvate. Second stage is the products in glycolysis, which are the 2 pyruvates are oxidized prior to the citric acid cycle. And the last stage is when the citric acid completes its job of oxidizing organic molecules and generating many NADH and FADH2 molecules. The cellular respiration is also done. So there you have it, cellular respiration.
Light energy is converted into chemical energy in a multiprotein complex called a photosystem. There are 2 types of photosystems, photosystem 1 and photosystem 2. They are found in the thylakoid membrane inside the chloroplast. These 2 photosystems absorb light energy through protein-containing pigments, such as chlorophyll. The light-dependent reactions begin in photosystem 2. In PS2, energy from sunlight is used to split water, which releases 2 electrons, 2 hydrogen atoms, and 1 oxygen atom. When a chlorophyll A molecule within the reaction center of PS2 absorbs a photon, the electron in this molecule attains a higher energy level. Because the state of an electron is very unstable, the electron is transferred to another molecule, creating a chain of redox reactions called an electron transport chain. The electron flow goes from PS2 to cytochrome B6F to PS1. As electrons move between these 2 photosystems, they lose energy. Because the electrons have lost energy prior to their arrival at PS1, they must be re-energized by PS1. Therefore, another photon is absorbed by the PS1 antenna. That energy is transmitted to the PS1 reaction center. This reaction center, known as P700, is oxidized and sends a high-energy electron to reduce NADP positive to NADPH. This process illustrates oxygenic photosynthesis, wherein the first electron donor is water and oxygen is created as a waste product. Cytochrome B6F and ATP synthase work together to create ATP. This process, called photophosphorylation, in non-cyclic photophosphorylation, cytochrome B6F uses the energy of electrons from PS2 to pump hydrogen ions from the lumen to the stroma. The energy released by the hydrogen ion stream allows ATP synthase to attach a 3rd phosphate group to ATP, which forms ATP. This flow of hydrogen ions through ATP synthase is called chemosmosis because the ions move from an area of high to an area of low concentration through a semi-permeable structure.
Light-independent reaction, or otherwise known as Calvin cycle or the dark reactions. This reaction does not really occur in the dark, but rather occurs at the same time with the light-dependent reaction. They don't require energy from the sun, so we call it light-independent reaction. The Calvin cycle occurs in the plant's stroma, which is a part of the chloroplast. The cycle has 3 phases: carbon fixation, reduction, and regeneration. First, the carbon fixation. The plant fixes a CO2 molecule into a ribulose bisphosphate, or RUBP, with the help of an enzyme, Rubisco. The RUBP is the start and end point of the cycle, that is why it is called a cycle. The RUBP becomes unstable because the CO2 molecule fixed to it, and that causes it to break into 2,3-phosphoglyceride. Second phase is the reduction phase. The ATP, which is a product of light-dependent reaction, will combine a phosphate group to the 3-phosphoglycerate. And then NADPH, which is another product from the light-dependent reaction, will give electrons, which will create 2-glyceraldehyde 3-phosphate, or G3P. G3P can be converted by plants into glucose, cellulose, or starch, which is essential to them. And lastly, regeneration phase. This basically converts 5 G3Ps from 3 RUBPs with some help of 9 ATPs and 6 NADPHs to produce again 3 RUBPs. Then the cycle repeats. The true product of this cycle is the G3P, as the glucose from it will be used as energy by the plant.
What is fermentation and what makes it different from cellular respiration? Fermentation releases energy from food molecules by making adenosine triphosphate when there is no oxygen available. It occurs after glycolysis. There are two types of fermentation, aerobic and anaerobic fermentation. Aerobic fermentation is kinda wrong since fermentation does not need oxygen in the process and aerobic means with oxygen. Well, the fermentation occurs like I explained earlier. The only difference is that the substrate in the process needed oxygen for reproduction.
Anaerobic fermentation is a method cells use to extract energy from carbohydrates in the absence of oxygen or other electron acceptors. This process follows glycolysis in the breakdown of glucose and other sugars to produce ATP. Through this, a cell is able to regenerate NAD+ from the reduced form of NADH. There are two classifications of anaerobic fermentation, namely ethanol and lactic acid. Both restore NAD+ to allow a cell to continue generating ATP through glycolysis.
That's all. Thank you.